Chapter 26 is about plant growth and development, and we'll get a chance to talk about plant hormones and how they play a role in helping these sessile organisms deal with the environment. So, sessile means that they are firmly planted to the ground and they can't move. Obvious. So, let's talk first about their early development. You've actually been watching these in your germination lab. The corn, the monocot, germinates in a different way than the bean example here, the dicot. Pretty soon we'll get to see the dicot. Let me start with that one coming out of the ground and the ones that we planted. And you've definitely seen this part. So the seed coat breaks down. This apical hook comes out. So basically, it's a tougher angle or tougher coating of cells because it in the soil conditions it's going to have to pull this heavy guy up out of the soil. So the roots grow down, this hook grows, and then this gets lifted up out, and then you'll see the cotyledons, which is where it gets its dicot name from because there are two cotyledons. These are called the seed leaves. They basically have a lot of nutrition stored in them, and that's what the plant uses to grow before your leaves come out and can do photosynthesis to provide energy for the plant. So these come up, and then you'll start to see the first leaves come out of that. And then as we watch some of our bean plants grow over the next week or two, we'll see that those little seed leaves wither away and go away, and then the new leaves come out. You'll get to see that. So the monocot, or the corn ones that we've been seeing grow, you've noticed grow a different way. There's the apex, or the shoot comes out of the top, and then the root comes out of the bottom. And I think we saw very nicely in a couple situations that the corn will right itself to be the right direction. So this is called a coleoptile, that first shoot that comes out. And we're going to see the coleoptile in some movement towards light experiments in a few slides. So that's how seeds germinate. You've done the experiments. You know that water is necessary to rehydrate all those nutrients that are in the seed and start feeding the embryo that's in there and allowing it to grow. I think this is the same picture. So let's talk now about hormones that help to control the germination process and also other growth times in the plant. We've heard probably about animal hormones. We haven't had a chance to study them in our sections yet, but we know that animal hormones can be peptides, it can be proteins, it can be small organic molecules. They are secreted from glands or cells. They're usually transported through the bloodstream. They're very specific, and they're regulated by our central nervous system. So that's what's in animals. We're actually going to do a little bit on that in a couple weeks. Plant hormones are a little different. They're small organic molecules. They are not peptides, and you'll see some structures in a minute. They're made all over the plant in different tissues. They can work either very close or they can be moved and around the plant to work in a distant location. They have a whole bunch of different effects, and meaning that one hormone doesn't just do one thing. It could do a couple things you're going to see. And their regulation is decentralized because the plants don't have a brain. They don't have something organizing their stuff like animals do. I split this slide into two, but there's a full page of it in your book. The hormones are listed here in alphabetical order, and that's not how we'll discuss them as we move on, so just want to mention that. You'll notice right away these are not proteins, right? These are our rings. We can start reminding ourselves about organic chemistry. Every vertice here has a carbon in it. This means there's a double bond between the two carbons. Remember, carbon has 4 bonds. Nitrogen's going to make 3. Maybe you remember your functional groups. So this is a C double bond O OH. So this is an acid. So oxen is actually indole 3 acetic acid. That's the fancy name for it. And remember that hydrogen might fall off, and that would be the acid form. Abscisic acid, again, that's a C double bond O OH. So this is a structure just like this I got hanging off of here. Brasinosteroids. Now this is a tough one, but you may remember the steroid backbone was 4 ring structures, and that's where this guy is somewhat related to. It's a very differently modified one. Okay, so just on this page that's discussing the structure. They're small organic molecules. What do they do? Abscisic acid, we won't talk about too much after this, but it helps to maintain seed dormancy, and it also promotes closing the stomata. So I like to think about this one as sort of a dampening hormone. Auxin we'll talk a lot about. Auxin was one of the first ones identified. It promotes stem elongation. So basically, it promotes cells to expand, not divide. Remember, plants can grow their vacuole and their cell wall can expand. It'd have to break down and grow, and the cells can get bigger and bigger and bigger. And you've noticed in your, probably even just your stomata lab, that you have cells of all different sizes in plants, and they can get bigger. They also, auxin is important in lateral root for initiation, axial bud outgrowth, leaf abscission. So this is when the leaf is going to break off, and then root elongation. For the most part, auxin is involved in cells getting bigger for plants to grow. Probably be a fair summary. Brassinosteroids, a lot of growth, promoting vascular tissue, promoting stem and pollen tube elongation. So another growth promoter. Here, these three, you notice that ethylene is the tiniest one. I'll get to this one last. Cytokinin usually works in concert with auxin. It's going to promote cell division. So where auxin will promote the cell to expand, cytokinin is going to pinch it off and make it divide. So they usually work hand in hand. So you see that they're effective at the axial bud growth, so the tips, and root growth, again the tips of the root to grow down more. Then jumping down here, gibberellins, a little more complicated molecule, just because it's got a bond coming out this way. Little 3D movement of that molecule you don't have to worry about. Gibberellins, another growth promoter, promotes seed germination, stem growth, fruit development, breaks the winter dormancy, mobilizes nutrients in seeds. So gibberellin is definitely a growth promoter. You'll oftentimes see this working also in concert with the cytokinins and the auxins. I saved ethylene for last. You're going to see a picture of the fruits that it affects later. It promotes fruit ripening. So it's ethylene as a gas, 2 carbons, 4 hydrogens, very small. It diffuses very easily. In fact, you can do an experiment at home. If you put a ripe banana in a bag, it's going to be exuding ethylene gas. If you put that with an unripe piece of fruit or an unripe tomato, perhaps, it will help that other fruit ripen faster. It's why when you go to the grocery store early in the peach season, they sometimes have paper bags there with your peaches, and they say put your peaches in a paper bag. The peach is emitting some ethylene gas, and by keeping it in the paper bag, you kind of keep it contained, and the fruit will ripen faster. They actually identified ethylene as a fruit ripening chemical or hormone by the fact that they used to have ripening sheds that they warmed up, and they thought it was the warming that did it, but they were burning kerosene gas, and one of the byproducts is ethylene. So essentially they were gassing the fruits, which promoted the ripening, and not the warmth had anything to do with it. So let's see some of the effects of these hormones. So both of these small plants are mutants that they know the gene that they shut down, and they know that if they put back the hormone, they can make, this one is sprayed with gibberellin, they can make it grow taller and the fruit fleshier. And then auxin definitely promotes long stem growth, you can see. Here's a picture. They always use grapes with gibberellin. Apparently our grape crops are sprayed with gibberellin. The plants take it up and make our fruit bigger and plumper. Again, growth promoting. Here's a picture of how gibberellin enhances seed germination. So this could be our, let's see, it's grown with a coleoptile. So this could be a corn. So water comes in and it starts to swell this endosperm. This is where the food is. Gibberellins are secreted by the embryo because now it knows that water is available. Gibberellin affects cells out here that make enzymes that break down the food reserves in that cotyledon storage area, endosperm, and that feeds the embryo so that you can grow. So that's one of the ways that gibberellins help break seed dormancy and help seed germination. So I told you that plants respond to light, and you've seen this. You've seen that a plant tends to want to grow towards the sun, and you've even seen this in the lab just now, that the roots tend to know which way is down, and they want to go with gravity. So closer up picture, auxin. If light is coming from this side, auxin is mobilized by the tip of the coleoptile. That's where it's made. So this is like a grass shoot. And auxin is mobilized down onto this opposite side that the light's coming from. and you can kind of see here it makes these cells longer. Where there's more auxin, it's made up here, transported down here. Where there's more auxin, the cells bend and get bigger, and that makes the plant lean towards the light. Similarly, the root is affected by gravity. Gravity is tugging on starch granules that are probably in the cells and some kind of plastids, and that causes the synthesis of auxin to be made, and then the auxin to be transported to the bottom side. I'm sorry, this is negative gravitropism of the shoot. I'm sorry, this is not the root. So again, it's going to the opposite side, and this promotes the shoot to grow up. So auxin is made on this side, it's transported to this side, these guys expand, and it forces the shoot to go up. The way roots sense the gravity, I don't have a graphic for. They actually think that one is these things called statoliths, which are plastids that are full of starch. They might be tugged down by gravity, and then there's another signal transduction pathway that makes the roots go down. But they're always a little sketchy on that in the other textbook, so maybe they don't believe that anymore, and that's why it's not in this textbook. That's just a real-life picture of how a plant, if you put it in a box and only have the light coming on one side, it definitely goes that way. You can just look out in your gardens and know that. Auxin is often called a molecule that causes apical dominance. It's high at the top. Remember, plants have indeterminate growth. They're always dividing. So the apical meristems have a lot of auxin in them. They tend to grow this way. Just look at how many slides are left. I think I might cut this off for a second here. So lastly, before we move, we take a little break and finish the rest of the hormones. Gibberellin and auxin, they're hormones, and they actually trigger a signal transduction pathway. Do I want you to memorize this? No. I'm just bringing it up to remind you. Remember, we talked about signal transduction in animal cells. Plants do signal transduction too. So here's gibberellin or auxin coming to the cell. It binds a receptor in the cytoplasm. That receptor gets dragged into the nucleus. This is kind of sounding like the steroid receptor story. It binds another transcription factor, which activates gene expression, and voila, the downstream effects are going to happen because genes get turned on. And that's the level of the summary that you want to know. I'm going to break here, and then we'll come back and talk about ethylene and then how plants sense light.